Alright, so today is restriction enzymes. Right. So the restriction enzymes are named according to the bacteria they're isolated from. So E. coli 1 is isolated from E. coli, and the biological role of restriction enzymes are for defense mechanism of bacteria, because in the bacterial world DNA goes from one cell to another, and when the DNA comes in from another cell, the bacteria has to cut it up, and it's going to cut up that DNA using the restriction enzymes, and it's going to distinguish itself by methylating its own DNA. And restriction enzymes cut at specific palindromic sequences, and we'll look at a picture to see what a palindromic sequence is. And the palindromic sequences are 4 to 8 base pairs long. And the restriction enzymes cut either in a staggered conformation or like they cut in a blunt end. We'll see that in the pictures too. Um, the site where it's cut, it's called restriction site, and so if you have four, so if the restriction enzyme is recognizing a 4 base pair site, then you have you have one cut every 256 base pairs on average. So you get that 256 because 4 to the power of 4 is 256, and then 4 to the power of 6 is 4096. So if the enzyme is cutting, if the enzyme is so the restriction enzyme that cuts using and recognizing 6 base pairs is gonna cut every 4096 base pairs, and the restriction enzyme that's going to recognize a 8 base pair sequence is gonna cut one every 65,000 base pairs on average. Um, and note, genome sequence is not completely random, so these are just on average. There's definitely a lot of different things that can throw it off. So these are all the different restriction enzymes: DiMH1, CLA1, ECOR1, ECOR5, HEND3, NOT1. So over here we have the palindromic sequence GGATCC, GGATCC, and palindrome here ATCGAT, ATCGAT, GAATTC, GAATTC. So these are all palindromes. And the cuts over here, see this cut's gonna give like a staggered cut, and this cut over here is gonna give a blunt cut. Um, so the test question is gonna be like, it's gonna be like a whole bunch of other letters here and a whole bunch of other letters here, and then he's gonna ask where is the sequence that restriction enzyme can recognize. And the uses of restriction enzymes in laboratory. We use it to identify DNA fragments and to identify mutations. Mutations we can see example later on, and changes in the palindromic restriction site will make it unrecognizable. We'll see the example of that in sickle cell later on. Um, and you can do restriction mapping. That's pretty simple. There's one slide on that. And southern blots, there's a slide on that. And you can use restriction enzymes to create gene constructs by cutting out different you can cut the cut the genes in different locations, and then ligate them using DNA ligase. So the phosphodiester bonds are being cut by restriction enzyme, and the phosphodiester bonds are being made by DNA ligase. So, and then once you have the constructs, DNA constructs. You can use them for transfect them into bacteria or mammalian cells, or we can make transgenic animals, plants, yeast, and bacteria. So this is gel electrophoresis and stained with ethylene bromide. And this is an example of using restriction enzymes, then gel electrophoresis, and then probing it with the radioactive probe. So first you have the cell, and then from the cell you take, let's say, all the human chromosomes, you cut them up into DNA fragments. So the human genome is 3 billion base pairs. So if you cut them with, let's say, a restriction enzyme that's going to cut every 3,000, after every 3,000 base pairs, you're gonna have, you're gonna end up with 1 million fragments, because 3 billion divided by 3,000 is 1 million. So then you take all these fragments, put in the slot, and then run the gel electrophoresis, and get the positive side over here, and the DNA is charged negative because of the phosphate backbone, the negative phosphate backbone. So the negative DNA is going to flow towards the positive electrode. And then if you have a million fragments, it's not going to look like these lines, it's just going to be a smear. And then once you have that, then you use radioactive DNA probe, like a insulin probe, and then you'll see which one of these lines correspond to the insulin gene, and then X-ray film shows you where it is. And the sequencing, we'll see how that's done later on. And this is a restriction map made with using restriction enzymes. So let's say this is ECOR1, the green one, and let's say the red one is not one. So let's say if you just use ECOR1, the green ones, you're just gonna have a cut here, and a cut here. You're not gonna have these red cuts. So this would be 1,000, and then this one would be 800, and then this one would be 1,300. So those are the only fragments you get. One, two, you'd get 3 fragments if you just use ECOR1. And likewise, if you use both, you get 1, 2, 3, 4, 5, 6, all these fragments corresponding with these. So you can make a gene map, restriction map, uh, by running the gel electrophoresis and then seeing how many base pairs long the different fragments are. So here, so if the gel is running this way, then these are the shortest ones, these are the longest ones, base pair wise. Maybe like you can say this is 6,000, this is 6,000 base, or this could be like 600 base pairs long, and over here it could be like 7,000 base pair long, and yeah. So then blotting technique, the procedure for that is, so you have the gel over here, you take that gel and you have the DNA fragments like this, and then you put the gel over here, got a tray, and you got a transfer buffer, sponge, and you got the gel here, and then you have the membrane, and you got paper towels and a glass and a weight. And then so the DNA is gonna get carried onto the membrane, because the membrane has an affinity for DNA, and then you're gonna use heat to fix the DNA onto the membrane, and then you're gonna put the membrane in a solution with the probes in it, DNA or RNA probes, and then they will hybridize to the DNA, and then you'll see the fluorescence or radioactivity and the probe. And then so out of all these, there were 2 that had complementary sequences in the DNA. And this is like looking for to see if there's any target DNA. So you have probe double strand, target DNA double strand, you heat it up to denature it to break the hydrogen bonds, and then they're gonna anneal, and the ones that have the right sequence they'll anneal, and these didn't have targets, so they didn't anneal, and this they anneal back together the probes. So if you want to, if you're doing like interspecies studies, or if you're doing gene duplication studies, then you can melt the DNA from two separate sources and then have them re-anneal to each other. So over here at 42 degrees, only the perfect matches are re-annealing. Over here, even though it's a little bit imperfect, it's still re-annealing because of the different temperature and that chemical. There's a chemical, and we don't have to get into that chemical. And over here, this is showing how they used to go around the problem of not knowing the sequence. So they used to know the sequence of the amino acids. And you don't want to use any amino acids like leucine or glycine or proline or arginine, where there's too many different possibilities for the triplet codon. You want to use something like tryptophan or methionine. And so then you know exactly what this is. For glutamate, you know it's either one of these two. For aspartate, you know it's one of these two. And then so you make a probe that's this one, and then you make a probe with this one. So you make 8 total probes because you have 2 times 2 times 2 times 2 times 2 times. So 6 different, so 8. So there's 8 total possibilities over here. 2 times 2 is 4, 4 times 2 is 8, so 8 different possibilities. So you make the 8 different probes. So like one possibility would be UGG, GAA, GAC, AUG, UCC, and whatever. And then the other one would be UGGAG, and then GAC, and then UUC. Another possibility UGAGGAC, and then that, and then UAC, and then and keep trying different possibilities. There'll be 8 total in this 20 because you need 20 nucleotides for a probe. And this one is looking to see if there are the same genes in different species. So you take a probe for let's say gene CORP1LR probe. So they cut up the DNA and then cut the DNA using restriction enzymes, and then they placed the probes, and the probes found it in one of the CORP genes in humans, and 2 copies of the CORP gene in monkeys, and they didn't find it in rat, mouse, dog, cow, rabbit, chicken, or yeast. So there's no CORP1LR gene in these. And then the KU86 probe was used, and it found that that gene was present in all these different organisms, different eukaryotic organisms. Um, so CORP1LR is primate specific, and KU86 has been conserved strongly evolutionarily. So, and also you can see that there are different sizes, the fragments. So this fragment's a lot smaller, this fragment's a lot bigger. And how to catch criminals using DNA. So you have the chromosomal DNA, and then you cut it up with restriction enzymes or restriction endonucleases, got DNA fragments, and then you put the DNA fragments, and then you run the gel, and then it'll separate into fragments, and then you add DNA markers, and you're gonna use like repeats or microsatellites and stuff like that for these. And then you're gonna compare the evidence with the suspects and the victim also, just to make sure you can find the victim's DNA. So then you have the evidence and then see where the probe attaches. So this probe and that probe and all these probes are exactly matching, the evidence is exactly matching this suspect, so he's guilty. And this suspect over here, they're not matching perfectly. See, this one is there, that one is there, but all these are missing, and this one is extra, these two are missing. But over here, this one, oh, these two are matching, this is matching, that's matching. So you can tell with a high degree of confidence that his DNA was found at the crime scene. And this slide is showing how a mutation can mess up a restriction site for an endonuclease. So sickle cell mutation, you have a T replaced with an A, and then the restriction enzyme cannot find it and it can't cut it. So in normal globin gene, you have beta globin gene, you have three cuts here, here, and here. But in sickle cell mutation, this cut does not take place. So what happens is you end up with a longer fragment. So the longer fragment is going to be 1350, and the shorter fragment is going to be 1150. So somebody who is homozygous for the normal globin gene is going to have this band, because they're going to have a shorter one because there's more cuts, so it's going to be shorter. And this one's going to be longer because there's no cut here, so it's from here to here, this whole gene is being stayed together. And in a heterozygote individual, one chromosome is going to have the sickle cell one, which is longer, and the normal one, which is shorter. And this one is hypersensitive sites. So we have two different cell types, erythroblast cells and the MSB cells. Erythroblast cells, you have an active globin gene because it's making globin. And so the globin gene is not wrapped around nucleosomes or histone proteins. So it's able to be cut up by the DNase, and the globin and MSB can't cut it with the DNase because it's wrapped around histone proteins or the nucleosome. And so then they cut up the genes with restriction enzymes, and then they used probes for the globin part, for the globin gene. And over here they found it at these concentrations was still present, but as the concentration of the DNA was increased, it disappeared, the globin gene, because the globin gene was cut by the DNases. And in the MSV cells, you always found the globin gene because they were wrapped around the nucleosomes and could not be cut. And so the probe always found it, and you can see it here. We don't have to worry about these. And that's about it. A little bit about northern blot. Northern blot is that they do not use restriction enzymes because they are working with RNAs, and RNAs are single-stranded, and restriction enzymes only work on double-stranded DNA.